Today we are going to discuss regarding structure of bacteria. Bacteria structure, ultrastructure of bacteria. Here we know that the bacteria is a microscopic in nature. It is very small. We can't observe by normal eye. We have to use microscope, compound microscope or electron microscope. We have to use for study of bacteria. If you observe the bacteria, it contains three layers. The outermost layers are three layers are present. So these three layers are first one is slime layer, second one is cell wall, and third one is cell membrane. Cell membrane or plasma membrane. So these are the three outermost layers. So let's start. First one, slime layer. It is also called as capsule. Slime layer or capsule. Simple. This slime layer is a gelatinous layer. It is a gelatinous layer on cell wall. It is a gelatinous layer on cell wall. So the chemical composition is generally polysaccharides, as well as murin also present. Polysaccharide as well as murin also present. Besides the polysaccharide as well as murin, if nitrogenous compounds or nitrogen compounds are present, besides the polysaccharide as well as uric acid, if nitrogen or nitrogenous compounds like ammonia, nitrogen are present, then it is called as capsule. It is called as capsule. So this is first outermost gelatinous layer on bacteria cell wall. Next one is cell wall. Next one is cell wall. We know that cell wall is outermost layer. It is outermost layer. It is rigid in nature. Rigidity in nature. Due to the stiffness or rigid nature of cell wall, it is give the particular size and shape to the cell. Actually, this is the main function of the cell wall. Cell wall is rigid in nature, or it is little bit tough in nature. So that's why it gives the particular shape to the bacteria. And as well as we know that the cell wall is made up of peptidoglycan. It is made up of peptidoglycan. The cell wall is made up of peptidoglycan as well as murin. Peptidoglycan. Peptid what? Nothing but proteins. So these proteins are a combination of amino acid. Next, glycol. Next one is a glycan. Here, peptidoglycan, it is a combination of glycan as well as proteins. Here it is peptidoglycan. So this is regarding what? Bacteria cell wall. Next, after bacteria cell wall, the third layer is cell membrane. Third layer is cell membrane. Cell membrane is made up of lipoproteins. Cell membrane is made up of lipoproteins. Lipo, nothing but lipids. Here, phospholipids and proteins. Peptide, nothing but proteins. Here, so this is regarding what? Cell membrane. Here, cell membrane, it is inner side of the cell membrane. It is going to separate the cytoplasm from cell wall. In between cell wall to cytoplasm, one small layer is present. Such layer is called as what? Cell membrane. So let's see the diagram, very small diagram of bacteria. See. So this is cell wall. On upper side here, on outer side, we are going to go gelatinous sheet. Gelatinous sheet. So that is called as what? Slime layer. And inner side here, one more layer. One more layer, such as plasma membrane. So this one is slime layer. This one is cell wall. This layer is cell membrane. Here, so this complete fluid, it is containing fluid. It is cytoplasm. Cytoplasm. Now we have to discuss external structure. Clear? Next. Here is cytoplasm. So first, cell membrane. Cell membrane, it is going to folded inner side. Cell membrane, it is folded inner side. So such a inner side folded cell membrane is called as mesosome. It is called as mesosome. Clear? Now first we are going to discuss regarding mesosome. What is mesosomes? Mesosomes are in folding of cell membrane towards a cytoplasm. So these mesosomes are equal to mitochondria. In eukaryotic cells. These mesosomes are equal to mitochondria in eukaryotic cell. So it means simple here is telling that true mitochondria is absent in bacteria. Clear? So that's why these mesosomes we can consider as mitochondria. What is the function of mitochondria? You know that mitochondria function is what? Respiration. It means here in mesosomes a respiratory enzymes are present. Respiratory enzymes are present. So that's why here aerobic respiration, aerobic or anaerobic respirations are takes place in cell membrane of bacteria. Next, mesosomes we can do into two types based on the location of mesosome. So some mesosome, some folding of cell membranes we can notice in middle of the cell. Middle of the cell. And some mesosomes we can notice lateral side. So simple. Central mesosomes, peripheral mesosomes or lateral mesosomes. The function of the mesosomes. The middle mesosomes that are involving in cell division, as well as DNA replication, Next, lateral mesosome involving in formation of septa, nothing but cell membrane. Clear? So this is regarding mesosome function. Clear? Next, regarding cytoplasm. So here this is entire fluid is a cytoplasm, inner side of plasma membrane. The fluid is present, that is called as what? Cytoplasm. First point, what I have to write here, generally in eukaryotic cell, the movement of cytoplasm we can notice that is called as cytosis. It may be clockwise movement or anti-clockwise movement. But in bacteria, movement of cytoplasm is absent. So it is called so here what we are going to write? Cyclosis. Cyclosis is absent. Cyclosis is absent. First part. Next, in cytoplasm, here the major difference between bacteria and eukaryotic cell, double membrane cell organelles are completely absent. Double membrane cell organelles. Double membrane cell organelles like mitochondria, nucleus, Golgi complex. So these all are absent. Second guy. And third, then which are present in cytoplasm? If these all are absent, then what are present in inner side of plasma membrane? Here, the cytoplasm is containing ribosomes. The cytoplasm is containing ribosomes. And the next one is ribosomes, and next one is one single chromosome, and next one plasmid. Clear? So this is regarding cytoplasm. The cytoplasm is a fluid. The fluid is a combination of water molecules, nitrogen substance, some you can say minerals, food molecules. So these all are present, water molecules, all are present in cytoplasm. Next, Cytoplasm is consist. First, we are going to discuss regarding chromosome. Chromosome. Here, in the first one is generally cells are two types. One is haploid cells, one is diploid cells. Haploid cells, they consist only one single chromosome. In diploid cells, homologous chromosomes. It means chromosomes are paired. So such cells are called as diploid cells. But here bacteria is a haploid cell. Only one single chromosome is present. Chromosome is only one single. And like eukaryotic cell, there is no prominent clear nucleus is absent. So here what we are going to write here, true nucleus is absent. True nucleus is absent. So it means chromosome is present in cytoplasm. So in such location where the chromosome is present in the cytoplasm, such is called as nucleoid. Nucleoid, nucleoid, or it is also called as genophore, PHORE, genophore, and it is also called as incipient nucleus. Incipient nucleus. The concept is very clear here. True nucleus is absent here. So that's why you can call it as open DNA or lacked DNA, you can call it. Next, one of the major important difference between prokaryotic cell to eukaryotic cells. So here generally in eukaryotic cell, DNA is made up of histogen proteins. But in this case, in bacteria, the DNA is not made up of histone proteins. Histone protein. So this is one of the major difference between eukaryotic DNA to prokaryotic DNA. In eukaryotic DNA, it is made up of histone proteins. In prokaryotic cells, DNA is not made up of histone proteins. Next, after chromosome, next one we are going to discuss regarding ribosomes. And you know very well what is the function of ribosomes? The function of ribosomes is, on ribosomes, protein synthesis is occurred. It is a factory of proteins production in the cell. It is a factory of proteins production unit in cytoplasm. So here ribosomes are mainly two types. One is 80S ribosome in eukaryotic cell, 70S ribosome in eukaryotic cell. Next, this 70S ribosome we can subdivide into two types. Subunits are, one is 50S, one is 30S subunits. Clear? Next, so ribosomes are in bacteria. 4 or 5 ribosomes have come together and bind with the mRNA. So such ribosome is called as polyribosome. Nearly 4 to 5 ribosomes are bind with the or attached to mRNA. M means messenger RNA. Generally, nucleic acids are two types, DNA as well as RNA. DNA means deoxyribonucleic acid, RNA means ribonucleic acids. Clear? So, such ribosomes we can call as polyribosomes. It is called as polyribosomes. Next one is first one chromosome, second one is ribosomes, third one is plasmid. See here one chromosome is present. It is a chromosome. So such portion is called as genome. And simple ribosomes 1, 2, 3, 4, 5 ribosomes like they are China or mRNA is equal to 5.1. These are polyribosomes. Next, plasmid. Beside the chromosome, it is extra or additional chromosome or additional DNA. It is double stranded circular DNA. But the replication is not dependent upon chromosome. It is a self-replicated information. The plasmids are self-replicated. These are self-replicated. So this is enough information regarding plasmids. Clear? Next one. Here, on bacteria, small granules are present. These are dark granules. These are food material. The storage food material is glycogen. The storage food material is glycogen. These molecules you can call glycogen. Plasmids are like double-stranded circular DNA. This is plasmid. It is plasmid. Next. So this is cytoplasm containing information. The complete double membrane cell organelles are completely absent, and it consists only chromosomes, ribosome, chromosome single, and ribosomes. We can call as polyribosomes. Next, plasmids, and storage food material is glycogen. Here. So this is information. Next, one more small information is here. This cell membrane, it is containing small channels. Such are called as porin. So this porin is a small channel which is useful for or allow the nutrition, allow the food molecules or nutrient into the cell. One small channel is present on cell membrane, which are called as porin. This porin is allow the molecules from outer cell to inner cell. Many nutrients, food molecules and nutrients are entered into the cell through this porin molecules. Next, and here In the last class we have been discussed flagella. From here cell membrane. Here to flagella cell. This is flagella. Regarding flagella, the last class we has been discussed. Flagella, it is arise from the basal granules of cytoplasm. And we already discussed in the last class some bacteria contain single flagella, two flagella, locotrichus, peritrichus, atrichus, flagella is absent, like it. Beside the flagella here, pili and fimbriae also present. Beside the flagella, pili and fimbriae also present. Pili and fimbriae generally are not useful for motility. Not useful for motility. Only flagella it is useful for motility. Not pili as well as fimbriae. See which are also arise from cell membrane. Generally, these pili or fimbriae are smaller than flagella. Smaller than flagella. Next, the pili is made up of pilin protein. So this is actually ultra structure of bacteria. Maybe the chances is there to ask 5-mark question answer. Very simple diagram, and this information is enough for ultra structure of bacteria. So generally, we have one small doubt here. Generally, sometimes when we are having of antibiotic also, for example, one person is suffering from fever, clear? Then we are going to use some antibodies. When we have antibodies also, sometimes the bacteria is not going to die, because the reason is only this one is, which one is slime layer. So this due to the presence of slime layer, it can have the capacity, it have the resistant power for some of the antibiotics. So that's why. So by using of antibodies also, we can't kill some of the bacteria. Why? Because gelatinous sheet is already containing resistant power to antibodies. Clear? So this is some of the information, exact 5-mark question we can use. Clear? Next. Now we have been discussed regarding structure of the bacteria. Now some more information is we are going to discuss. That one is nutrition as well as reproduction of bacteria. First one is nutrition. Nutrition of bacteria. In the Bacterial classification, we have been discussed. Maximum types of nutrition are shown by bacteria. Maximum type of nutrition, more than them, it can show. Generally, nutrition is mainly two types in bacteria. First one is autotrophic. Next one is heterotrophic bacteria. Autotrophic bacteria as well as heterotrophic bacteria. Autotrophic bacteria, the definition I hope you already know, because same definition for photosynthesis. Autotrophic bacteria we can divide into two types. The first one is chemo autotrophs. Next one, photo autotroph. Photoautotrophic bacteria as well as chemoautotrophic bacteria. First, what is autotrophic bacteria? Auto means self. So some of the organisms that have the capacity to prepare the food molecules by its own mechanism. So that is known as generally photosynthesis definition. Example is all green plants. All green plants that have the capacity to prepare their food material by its own mechanism. The method you know that is a photosynthesis. In bacteria, autotrophic nutrients are first one is photoautotrophic, second one is chemoautotrophic. The definition is there already in this word. First one, photoautotrophic. Photoautotrophic. Photoautotrophic means what? Here, some of the bacteria that contain bacterial chlorophyll. Bacterial chlorophyll is present, and also, and some of the bacteria are containing chromatophore. So generally these two are equal to chlorophyll of chloroplast. So simple. I hope you got the point. These two are equal to chlorophyll. The chlorophyll have the capacity to capture the sunlight. The sunlight is get converted as chemical energy, and the chemical energy is useful for preparation or storage of organic substance. Clear? So these are what? Autotrophic, photoautotrophic bacteria. Example: green sulfur bacteria. Green sulfur bacteria. Next one is chemoautotrophic. Chemoautotrophic. Again, the definition is there in this heading. Chemo means chemical reactions. Here, these bacteria they prepare the food molecules by using of energy. This energy is coming due to the chemical reaction or oxidation of inorganic substance. Here, in chemoautotrophs, during the oxidation reactions, oxidation of inorganic compounds, At the time, in these reactions, they are going to release small amount of energy is released. So such energy is enough to prepare the food molecules. So such type of bacteria we can call as chemoautotrophic bacteria. Example is iron bacteria, sulfur bacteria, iron bacteria, sulfur bacteria. So these are examples. Next, heterotrophic. You know that already. Heterotrophic means what? Such organisms that have no capacity to prepare the food molecules by its own mechanism. They should be dependent upon another organisms. Again, it is two types. You know already very well. First one is saprophytic, and second one is parasitic. Only one word is enough, these two definitions. Saprophytic. So these organisms, some of the microorganisms, they obtain the food molecules from dead and decaying organisms or tissues. Parasites. These obtain the energy or obtain the molecules from living organisms, such as well as parasitic nutrition. Simple here, they obtain the nutrition from dead. They obtain the food molecules from living organism. This is enough. So this is regarding what? Nutrition of chromosome. Nutrition of bacteria. Next one is reproduction. Reproduction, again 3 types. First one is, you can write directly 4 types. One is asexual reproduction, one is sexual reproduction. Asexual reproduction by binary fusion, in the bridge course, we have been discussed difference between asexual reproduction and sexual reproduction. In sexual reproduction, there is no formation of gametes, there is no fusion of gametes. Zygote is not formed in asexual reproduction. In sexual reproduction, fusion of gametes occur. Clear? Binary fusion as well as endospores and also. Next, sexual reproduction. Sexual reproduction, we can divide it into 3 types. The first one is transduction, next transformation, next conjugation. First one, transduction. First one is transduction. Here, in this, generally the thing is here, sexual reproduction, it is not a true. It is not a true sexual reproduction. Why? Because in true sexual reproduction, male individual is going to produce male gamete. Female individual is going to produce cell. Both are fused. It is called as fertilization. Fertilization to zygote is formed. But in this case, gametes are not formed. Clear? So that's why it is not a true sexual reproduction. It is called as parasexual reproduction. Parasexual reproduction. First one, transduction. So in this case, generally here, transfer of DNA material from one bacteria to another bacteria with the help of vector. So such is here bacteriophage. Nothing but virus. Nothing but virus. Simple. What is the definition for transduction? In this case, what happened in this method? Transfer of genetic information, nothing but DNA, from one bacteria to another bacteria. But for transfer of genetic information, which is required? One virus is required. The name is what? Bacteriophage. So such process, such method is called as transduction. Next one is transformation. Simple here, if you kept 2 bacteria in one medium, one is going to release the DNA into the medium, and the second one is going to receive the DNA from medium. So simple, it means what we are going to write here? Transformation means bacteria received the genetic information from external side or from the medium. So that is called as what? Transformation. The bacteria receive the genetic information about DNA from environment or external medium. Next one, maybe. Next, conjugation. So in this conjugation, two different bacteria we can call as strains, plus strain, minus strains. So these two bacteria come together to form conjugate tube is formed. Simple. This is our bacteria. This is our bacteria. The half will consider this one is plus, this one is minus. Plus stain, minus stain bacteria. When these two bacteria come together, here one connection tube is going to form. Connection tube. Nothing what we can call as conjugation tube. After formation of conjugation tube, this is a DNA, male, this is a female. From male, some little amount of DNA is transferred from male stem to female stream. So such is called as what? Conjugation. Clear? So this is an entire discussion regarding bacteria. So if the question is asking only regarding write an essay on bacteria structure, then no need to write regarding nutrition as well as reproduction. Clear? Just you have to write one small diagram and you can see cell wall, slime layer, cell wall, cell membrane, cytoplasm, ribosomes, next nucleus, and food material is glycogen. So this information is enough, and along with the diagram. Okay? So in the next class, in the next video, in the next session, we are going to start archaebacteria,